I'm Spencer. I'm a microbiologist, kind of card carrying microbiologist by training. I did my PhD at UC San Diego, and I worked on cyanobacteria because I was very interested in kind of carbon capture by cyanobacteria and how they metabolically function. And so my whole career has been there's been a lot of different threads of green technology and climate change related science. When I came to Berkeley to do my postdoctoral work, I worked in the lab of Jill Banfield, who developed this technology that I'll kind of touch on in this little presentation today called genome resolved metagenomics. And I don't know how many biologists or biology uh, uh, tangential people do we have in the audience. So, okay, cool. So like most, I'll try to keep it like light on the super technical details, but like feel free to ask questions. But yeah, like Jill basically developed this approach to go into microbial ecosystems and sequence all of the DNA of those ecosystems. And that can tell you basically what microbes are there, what their functional capabilities might be, and potentially who they interact with and like why microbes in, as a group, as a collective, are doing certain behaviors in systems like soils or wastewater treatment plants that, we can observe, like produce methane or produce CO2 or produce nitrous oxide, but like how is that actually mechanistically happening like in a collective set of microbes all kind of working together. And so, we've taken that approach and I've started my own lab here at the Innovative Genomics Institute just to also give a little background on the institute. It was founded in I think 2015 by Jennifer Doudna. That's the some person who uh, was had some part in developing CRISPR-Cas editing technology. She's kind of our boss, I guess you could say. But ultimately, she her vision was that CRISPR can be used for good, right? And so the institute focuses on applications of CRISPR technology to human health, to climate change, and just overall to the betterment of society as a whole and the planet. So here, hold on. I'll share my screen. And kind of what I'll tell you guys about today is a really big project that we're working on where I'm just, oh, does it have the slide view or the presenter view?
I have the slide view.
Okay, great. Okay, then let me find my cursor here. Okay. So yeah, so basically, I'm part of this very large project at the Institute called the Audacious Project. And a very large portion of that project's money, which is $70 million dollars over 7 years, has been allocated to effectively finding gene editing solutions to mitigate microbially mediated climate change, particularly the release of methane from ruminant animals. So, if you're not aware, cows make a significant contribution to global methane release. And if you look at kind of global methane sources from kind of natural systems, this is exclusive of kind of methane release from industrial processes, which makes up about, I think, like 30 or 40%. But most methane actually comes from biological sources. And most of those biological sources are microbially mediated. So, like the microbial activity within landfills or the microbial activity in agricultural soils, particularly rice, and in the rumen of livestock. And so, I mean, I kind of got the crash course in the cow in the last year because that's when we started kind of really working on this project in earnest. I didn't know much about cows before that, other than at least I eat meat, and so I think they taste good. But they do have this really miraculous organ inside of them, which is basically when you hear about cows having multiple stomachs, it's like their first stomach. And this stomach or this compartment is called the rumen, and in there they basically have a unbelievable anaerobic fermentating bioreactor that you can put in kind of any organic material and it transforms that material into volatile fatty acids, which are making up a huge portion of the animal's energy needs, and then ultimately also producing methane as a waste product, which is expelled mostly through the mouth as burps and only a little bit as farts. So if people say cows fart a lot, they're actually wrong. They burp a lot and they expel methane that way. And so, you know, because this process is microbially mediated, we're of the mind that it's actually this. What we really need to know is how does the factory work, right? Like if you have a factory that builds cars, there's a bunch of different independent machines inside that factory that each do little tasks, and they all kind of work together to produce a car. But maybe that factory is not working at the highest efficiency possible. So when methane is released from cows, you're actually losing about 12% of the energy in calories that's being taken in by the animal just to that loss. So if that energy could theoretically be redirected into things like volatile fatty acids or other byproducts, that could be more add more efficiency to farming and ultimately reduce greenhouse gas emissions. So kind of win-win situation. And a lot of times, I've given this talk to people and they're like, well, why don't people just stop eating meat? And I'm like, well, that would be a solution. If we eliminated all the methane from cows right now across the earth, then we'd be in a much better climate change position. But the truth of the matter is developing economies are probably not going to stop eating meat, and there's a huge amount of livestock that's used for work across the world. So water buffalo and cattle, things like that are still used for plowing fields, and the numbers are quite substantial. So, as I said, IGI is kind of the home of this technology, genome resolved metagenomics. It just turned 20 years old. And the importance, kind of the way this technology works is you have a bunch of microbes in an ecosystem, like bacteria, viruses. There's little segments of bacteria called plasmids, which exist inside bacteria. These things all are existing together, and we just basically extract all of the DNA from a sample and shear it up and sequence it into tiny little chunks of 150 letters each. We then assemble those sequences into longer pieces called contigs, and then we associate those contigs with each other to reconstruct the genomes of these bacteria that were present in the original sample. And then we can do subsequent analysis on there. And basically, the information you're getting from this is like who's there and what are their metabolic capabilities and how much of them are there, right? How much of each individual is there. And so the real importance of this is that only 1 to 10% of microbes have actually been cultivated in isolate. So the rest can really only be detected using DNA sequencing. And it's kind of rewritten our understanding of the microbial tree of life and taxonomy and function. And just to kind of give you a sense of like what that looks like, what you're seeing here is a picture. It looks very complex because it's the entire metabolism of an uncultivated microorganism that grows in soil. So it's showing basically like all of the different metabolic pathways that are occurring inside this microorganism. And the way we were able to infer this was simply by looking at its reconstructed genome from genome resolved metagenomics and getting a sense of, how is it producing energy? Like it has electron transport systems, it has, glycolysis, TCA cycle, it has like a bunch of different functionalities that, we might find in a microbe, but, we can kind of put it all together instead of, not knowing what it does because we can't cultivate it. We can know something. We can also link functions and interactions to outcomes. So, like, this is some data from experiments that I've been running in my lab in collaboration with UC Davis. And we basically are looking at if we cultivate rumen microbiomes in the lab, can we identify metabolic functions in these genomes that are growing in these microbiomes that are associated either positively or negatively with methane production from these microbiomes? So we can measure how much methane comes out, and we can correlate that to kind of the metabolism that's occurring inside the system. And we actually can find certain types of metabolism are more or less correlated. And, obviously things like methanogenesis are highly correlated with methane output, but just to give you kind of like a picture of that. So, where does this all go ultimately? And what's this kind of project all about? Well, we Jill, my former PI, and Jennifer kind of had this vision. Like genome resolved metagenomics is giving you the ability to see the genomes of all of these microbes that exist in these communities that are doing important things. And CRISPR-Cas targeted editing technology is giving you the ability to basically make precise edits anywhere in DNA sequence that you know. So if you know the DNA sequence of something, you can target, and edit to that spot. And so it kind of makes sense that these two technologies, go hand in hand if you want to edit microbes that are in a complex community setting. And so myself and two other researchers here at the Institute are also now principal investigators, Dr. Ben Rubin and Dr. Brady Kress. We together with Jill and Jennifer developed a methodology for targeted editing of microbial communities. So basically, like if we have, let's say, a community of microbes in our guts, and that community is making, some inflammatory compound, we can identify that who's in that community, what their genome sequences are. We can then determine if some of those microbes are editable, and then finally we can deliver a targeted editor such that it could, adjust. The hope is to adjust and shift communities in ways that then, maybe it wouldn't produce that community wouldn't produce that inflammatory compound as much. And to kind of illustrate that of how we're thinking about doing that in cows is number one, this is really where I come in, more of my expertise is modeling microbiomes. So if we look at the rumen, can we start to understand kind of the trophic structure? Can we understand when complex carbohydrates come in, who's first responsible for like breaking those down? And then who does the secondary fermentation? And then ultimately, who produces volatile fatty acids or CO2 as a byproduct? Who might produce hydrogen as a byproduct? And then you can see that there's kind of this at the end point, this competition that goes on where there's a lot of hydrogen in the rumen, but it's getting consumed by organisms that produce acetate or other volatile fatty acids. It's also getting consumed by methanogens to produce methane. And so we want to kind of, shift that, right? So how do we, if we understand how the factory works and we understand all the parts and we understand all the genes that are involved, can we develop a strategy where we edit, a certain number of microbes or certain genes within specific microbes that would shift that community's functionality away from producing more methane towards something like producing more volatile fatty acids. So like that you're getting more efficiency and the cow's getting more, calories out of what it's eating. And so we're modeling these microbiomes, we're developing and testing these editing strategies like in vitro. So we do this in the lab. And then potentially, at the end of this project, we're hoping to develop delivery strategies so we could take these DNA editing technologies, potentially encapsulate them in a pill of some sort or, an injection, and then you give it to the animal. And then this is a permanent change. So the hope is that we're doing like really rational, specific, predictable, and permanent editing to microbiomes. And so this is kind of like a platform technology. Like if we can do this in cows, we could do it in people, we could do it in soils, we could do it in, wetlands, like wherever we need to shift microbiome functionality in kind of whatever direction we want. I mean, this is really the future of kind of microbial engineering. Because right now, there's a lot of genetically engineered microbes that are doing things like producing insulin for people, right? But that's just a singular microbe in a factory that's being grown in a huge quantity. I mean, what if, you could leverage all of the power of and kind of diversity of an entire community of microbes, which ultimately is how microbes like to grow, right? So we need to kind of understand and how to manipulate them in their natural contexts. So there's still some big barriers, right? Like, so we still need to know how to cultivate microbial communities better. We need to understand how to model them better. That's stuff that my lab explicitly works on. Um, we need better genetic tools for them. So like right now, we have a ton of genetic tools that let us modify E. coli and a lot of other bacteria that you can, read about in the news, like salmonella, generally things involved with human disease. But we don't have a lot of genetic tools to modify bacteria that are in the environment. And that's what we need to develop. And then, the final thing is ultimately like delivery strategies for this. Like, so how do we, if we know how kind of what to edit, we can edit it and we can show that that kind of works in a contrived system in the lab. How do we send that into an animal or a location and have it kind of, work? And just to put this in perspective, I am one part of a very large team involved in kind of the technology development and tool development for this. And then ultimately we have teams involved in formulation, doing field and clinical trials, and very importantly, outreach. So we have a team explicitly dedicated to kind of understanding the policy side of this because there's really no regulation on like if you created a product today and said, I have a pill that can modify your microbiome and I want it to be like an FDA approved medicine, there's like not really good, policy avenues for how to regulate that. And so we're heavily involved in like informing the government about, like what we're thinking about doing so that they can develop regulations and policy around what we're doing. So that's really what Manar and Melinda spend a lot of their time with. And ultimately like that feeds back on us. Like the government may say, we don't think that, this is a safe thing to do, or, we'd prefer if you used, this modality to do editing. And so ultimately we don't want to waste our time or people's money doing something that, that the government might have trouble regulating, right? And like we want it to be safe. So just to give you like an example of kind of like a study that we're currently working on and like how we get to these answers. Um, Basically, it all starts with kind of discovery of targets in the rumen for like reducing emissions. Like, how do we identify what we want to edit? And this is just showing on the left, like a picture of the quote unquote hydrogen economy in the rumen. So, like, basically, a bunch of feed goes into a cow's rumen, and then a bunch of like digestion happens, and ultimately, you end up in kind of this middle space with a lot of hydrogen and CO2. And this can go in like a lot of different directions, right? So it could be utilized by acetogenic bacteria to make acetate, which is a volatile fatty acid. So that's a good thing. Could be used by fumarate reducers that produce succinate, which is also a good thing. It can also be utilized by methanogenic archaea to produce methane. And then kind of like that's an endpoint product. Nobody really uses, no microbes use methane in the rumen, so it's just released. And if it's not released, you can have problems. So ultimately, like you need this energy flow through the system to keep the system running functionally. So methanogens actually serve kind of important purpose in that they're kind of at the bottom, they're like the drain at the bottom of a pool, kind of like pulling the water out, So it's like it drives the system forward. And so what we did is we ran an experiment where we gave cows an additive, which in their feed, which is a red seaweed, and these red seaweeds are known to suppress methanogenesis in these microbial populations. So like they contain compounds that suppress methanogenic activity. And what we wanted to know is when we did something like this, how was hydrogen redirected? So we're kind of using, even though this is these seaweeds are being investigated as a potential feed additive to sort of reduce methane production from, cattle in kind of feedlots and stuff. One of the problems is most cattle graze, so you can't really like feed. Only 1% of cattle are actually in a situation where they're being fed every day. Um, and so feed additives are great for, kind of high density agricultural systems, but they're not great for like the majority of a cow's life when it's just grazing on the range. But we're using it in this case as a model to sort of look at how do if you're kind of removing the methanogens from the situation, like what is the response of the microbiome in that case, and can that teach us something about organisms that may be able to fill that niche if methanogenesis is removed, or like could be potentially competitive with methanogens, and like why or how could they be competitive? So this is just showing we're able to identify one out of these kind of, this is just showing a plot of the abundance of various different microbes in the rumen after this treatment. And all of these microbes that you're looking at, they all have the capability to utilize hydrogen as an energy source, right? And this one particular microbe, stood out, and it was like really abundant in the samples where, we treated the cows with this red seaweed. And because of our ability to do genome resolved metagenomics, we could actually look at the genome of this microbe and predict functionality that it had. And what, to sort of summarize this, ultimately, this microbe had a number of different traits and functions that like made it competitive with the other hydrogen utilizing microbes. So like it had a way to sort of bring in alternative substrates like aspartate and malate and transform them into fumarate, which is what it uses to couple its hydrogen utilization to. So like it basically has metabolic properties that gives it advantages over other hydrogen utilizing organisms. And so that's like kind of what leads us down the path of thinking about well, maybe we could utilize some of these particular genes and strategies that this microbe uses to make others or even this individual microbe more competitive against methanogens. Like maybe we could enhance its ability to uptake aspartate and malate. But this is how we sort of like go down the path of understanding what knobs to turn in the system to ultimately shift the system in a different direction. And so yeah, so I think just a couple concluding remarks. So ultimately, like we're very focused around this fermentation from the rumen producing methane, but really this is kind of a whole bioeconomy. Like the rumen is ultimately going to produce feces, which end up in these like waste lagoons around cattle farming locations, and these waste lagoons produce their own methane and their own and nitrous oxide from the degradation of nitrogenous products. Also, sometimes this waste is then put into anaerobic fermenters to produce biogas. So actually, the process is working in reverse. You actually want more methane in these conditions, but actually understanding the rumen and all of the microbes that are present there teaches us a lot about what's happening in an anaerobic fermenter. And so ultimately, there's the outputs and inputs to these systems are significantly impacted by their own and preceding microbiomes. So like microbiomes are really kind of driving all of this stuff that's going on, and the microbiome that exists in the rumen is going to impact the microbiome that's in the waste. And a lot of these systems share similar microbiome structures. So learning about one is kind of broadly applicable, and potentially altering the structure can significantly increase our efficiency. And so with that, kind of like I'll thank you guys for having me. And I hope it wasn't too technically detailed, but if you have any questions, I'm happy to answer them.
Yeah, that was awesome, Spencer. Thank you. Um, I know I'm gonna see if there's a better way that you can see everyone, but I know I have a question.
Well,
So
stop. Okay.
is the idea that like if you were to give it a like a pill or something, would you be able to like then pass down vertically, so like mom to child, the edits that you made, or is that something that you would have to keep kind of giving them a treatment at like at certain stage in their life?
That's an excellent question because one of the things that kind of came up at the start of this project was we all sat down, and one of the first questions we kind of asked each other was like, does anyone know? Because we're also like not rumen biologists, like the people at IGI. We then kind of got together with people at Davis who know a lot more about rumen microbiology, but they don't, know as much about genomics and kind of like they hadn't really thought about the communities of microbes in ways we do. And we sort of sat down with them and said, how does the rumen microbiome develop? Like, does anyone know? Like when cows are born, when do they start making methane? Like, what is, what's the first microbes to colonize them? Are they sterile? Like what's the deal, right? Like because we hear a lot about, natural birth in humans versus cesarean section, and like are you missing out on, like vaginal microbiome components that are very important for the developing baby, and we really just don't know that for cows. And so what we actually did as part of this project, a huge component. So I showed a picture of Dr. Rohan Sachdeva, who's kind of like one of my closest confidants here at the institute. He's running a study right now with UC Davis where basically we have groups of, I think, 8 or 12 cows, and all we're doing is they're being born, and we're sampling the mothers, and then we're sampling the cows, every month as they grow. And basically, there's some of them that are just receiving no treatment. Some of them are actually receiving some of this methane suppressing additive, like right from birth until death. And then some are receiving it just for a period of time in their early life, and then it stops. And so we're assessing both kind of the microbiological development of the rumen, whether that's linked to the mother's microbiome, the surrounding environment, and then also, can that microbiome be altered throughout the life of the animal by kind of suppressing methanogenesis right at the beginning? So I think that was a long answer to your question, but ultimately it's a great question because it's something we are particularly curious about. And I'm most definitely investing a significant amount of time and resources into trying to understand.
Thank you.
Yeah.
Yeah.
Um, I guess it's kind of similar to the last question where you were explaining, but would the rumen microbiome differ based on like where the cow is in the world? And how is like have you thought about how this can be like accounted for with like the pill and what you're targeting, or how expandable is this?
Yeah, so that's another great question. So there have been kind of broad surveys done on rumen microbiology that can give us a sense of like how much diversity is there. So people have taken like thousands, like I would say there's in the thousands of samples that have been taken from adult animals across the earth, right? And kind of from certain major locations that are interested in ruminant, farming. Like so you and ultimately what you see is that the methanogenic population, so the populations of archaea or these, non-bacterial but kind of look like bacterial organisms that produce the methane, um, they're they're reasonably stable. Like there's actually not a huge amount of diversity in those species, but the bacterial populations are widely variable. So there's actually a huge amount of diversity. I would say that we probably not even scratched the surface of the potential diversity that's in the rumen. And it's quite interesting though, because if you look at it from a functional perspective, there's a lot of organisms that do the same thing or that do combinations of the same thing, right? So you always have organisms that can degrade complex plant fibers. You always have organisms that can ferment those sugars that have been degraded out of the fibers into volatile fatty acids or lactate or like a number of other things. And you always have, methanogens and other types like hydronotropic organisms. So thinking about it functionally, we should, even though there's a large diversity in the microbes, and yes, actually, some other excellent work that's been done has shown that, like, kind of like what are the main determinants of variance, right? Like, what are the main drivers of rumen microbial diversity? Like, one of them would be diet. I think that's a huge one. And another one is location. And then, yeah, and genetics actually, interestingly, doesn't play as much of a role. So it's kind of like mostly diet and location is like kind of the main factors driving it. And so, yeah, I think so. To answer your question, like, how would we do this kind of across the diversity of ruminant animals? Well, I think we're thinking of it more from a functional perspective than from a like a species specific perspective. So like the ideal situation, because yeah, that's a great, it's great to think long term. Like, right, we can do this in a dish, but then how do we apply it, across the diversity of animals? Well, I think the starting point is you kind of start with a population of animals that might be like, more local and you kind of understand better. And then ultimately where we'd want to go is we'd say, okay, maybe here's a cocktail of things that is going to hit 80% of animals, or here's a cocktail of things like there's certain microorganisms that are present across many rumen microbiomes, right? There's like kind of conserved core genome or conserved core species, right? And so maybe those ones are good targets. Um, and I think that's a lot of what we're thinking about in the long term. But yes, I think ultimately we need to design CRISPR guides that are going to target specific loci. And unless those loci are conserved across like a large number of microorganisms, we're going to have to design a lot of guides. So yeah, I think that it is going to be a challenge to sort of hit everything very, very broadly. But I think that in the near future, if we're looking at a specific herd and we kind of understand that herd well, that's kind of the starting place. But that's a great question. Yeah.
So if, I mean, it's a big problem now. Something like, I guess first let me understand, like, for this editing, are you would be targeting something that would kind of divert away kind of the flow to producing methane. So would it be targeting like a specific enzyme in a methanogen, or would it be just trying to kill them? Or
Right. Yeah, I think so. We've discussed a lot of different. So, right, from the start, we kind of had some amount of understanding of like where we'd want to go. Like, there was, the reason I brought up that hydrogen economy picture was there's been a lot of room in microbiology done, particularly at the macro level, to kind of understand like, oh, there's sort of these different groups of microbes doing different things in the room, and we may not know like every individual species and all their genomes and everything they're doing, but we kind of have a bulk picture of like where the energy flow goes, right? And so hydrogen, I think, is definitely a target. Like that's any organism that's utilizing hydrogen is that's not a methanogen is ultimately a competitor with methanogens, right? And so there's a couple of ways that we can go about, reducing methane output from the rumen. One being, directly attacking methanogens. Like we could, you know, one of the proposals is like, could we design a phage, or a archaeal virus that would be able to kill methanogens and specifically kill the ones that, we don't want. Um, other proposals have, focused around could we, restructure the microbiome in a way that there's strong competition against methanogens. So like give advantages to organisms that will compete directly with methanogens for hydrogen or for other products, and then ultimately push them out of the system and allow the system to still have a sink for those electrons, right? So you need to hydrogen. If you don't use the hydrogen, like when you actually get rid of methanogens, cows start burping up lots of hydrogen because it takes time for other microbes to fill that niche and utilize that gas as an energy source. And so ultimately, there's still energy loss, right? So we want what we would ideally like is this sort of win-win situation where we reduce methane production and we also increase efficiency, right? Because I think farmers a lot of what I've done is actually go talk to farmers. I'm like, well, kind of like, what are you guys interested in? Like, what do you want? And they're like, well, obviously, we want to, here in California, there is some degree of, I think there's some degree of carbon credit system, but one of the issues is like, it's really hard to quantify how much carbon you should be credited with. So that's a whole other discussion. But ultimately, I think, farmers are interested in being more green, quote unquote, to save to get these carbon credits, but they're also, obviously interested in being more efficient. Like they want efficiency equals more money for them. So I mean, ultimately, I'm personally not, interested in helping like large corporations make more money, but I am interested in efficiency gains that, help the climate crisis. So I think that's sort of like where we kind of derive a lot of the strategy from is we're thinking about, what types of modifications could we make that are efficiency increasing, but also methane decreasing. And we're still kind of at the very beginning of this, so a lot of it's sort of a discovery phase of figuring that out. Sorry, again, that was a long-winded answer. I hope that answers the question.
thank you.
How do you think your approach will shift when you're speaking to more conservative farmers? I mean, in terms of politics, like when you're going to the Midwest and stuff.
You mean my approach and like how I give this presentation?
Mm-hmm. Yeah.
I mean, I think, I think one of the things I've learned in science communication is like you want to find a common ground, right? And I think for a lot of more conservative individuals, that common ground is money. And like it's like the bottom line. Like for instance, the people are like, oh, oil companies might be like sea level rise like isn't real. Yet they understand that if they don't raise their, oil rigs on the ocean, like they're gonna lose like some multi-billion dollar investment. So then they raise them, right? So I think that, explaining to people that ultimately your bottom line is going to be hurt if you don't embrace new technology, that I think gets through to people. Like, we're not coming here to sell you, some California hippie crap. We're coming here to sell you something that's going to make you money. And I mean, in the end, it's the right thing to do, right? Like, I mean, I don't know. Ultimately, also, I don't know if this our particular technology is the answer. It's just one avenue, right? And understanding microbiomes better is going to help us, do like maybe it's not like editing, but it's just like maybe we'll learn something where we'll say, oh, actually, we need to just increase the amount of, glutamate like in the diet, and then it's going to be better. So, um, yeah, I think like that the learnings from this stuff is just what's important. And I would say like ultimately, you know, im implementing these types of efficiency increasing changes in your strategy is how you're gonna, move into the future and like be ahead of your competitors. Yep.
Maybe it's really interesting to learn about the different steps of what you're thinking about and trying to determine which microbes to edit. How do you determine which ones you want to focus on editing? Are there certain that are easier to edit than others, or is it just looking at what you're talking about, like looking at hydrogen versus other things?
Right. Yeah, that's a great question. So I think there's kind of two that one of the things that I get approached by. So we have kind of like my team, which is the discovery team, and then there's like an editing team, right? And the editing team comes to me and they were like, "What should we do?" And in the this is the second year of the project out of 7 years, and so it's kind of like we're at the point where I have some answers, but in the beginning I had like none. I was like, I'm not really sure yet. So, um, but I think we've kind of broken it down into sort of two columns, right? In one column, you have functions that we really, really would love to try and edit, right? Like we're like, oh, this hydrogen utilizing microbe has a protein, a hydrogenase that allows it to like consume hydrogen. Let's like start tinkering with that and like let's like put that into different microbes, right? And like maybe we can optimize it. But like I can just tell you from, tangential experience that like working on hydrogenases is like really hard. So, that's sort of like a really want, but it's really difficult, right? And then in the other column, we have like what microbes are easily accessible, right? Like what microbes could we edit with our current tools now? And so there's this intersect, we kind of, do a little intersection of these columns and try to figure out like, okay, if this microbe is editable right now, what types of changes could we make in it that, might be really desirable targets or at least close to desirable targets. And to full disclosure, I would say that kind of like even doing this at all is really cutting edge. Like I'd say, the ability to edit kind of any microbes in the room in a community context, like if we do that in the next year, it'll be like one of the first times that it's been done. So, or at least I think it would be the first time that it has been done in the room and in a community context. So it's kind of like it's like taking that first step onto the moon, So, um, so I think what we will start with is things that are easier. So I think we kind of lean towards the easier category, and then once we have a handle on that, we'll go more towards the relevant category, right?
So, I mean, thinking about like the easier stuff, do you find that like you're leaning towards more subtractive things? I mean, I guess you would be if you're trying to knock out methanogens, but like, do you think there's an opportunity for kind of additive, so like add functions to microbes that aren't already there?
Personally, I think that the subtractive strategy is not a good strategy. I think that like ultimately, if you subtract something out of microbiome, like something will just fill its place, right? And what you need to do is you need to add function so that you can push the microbiome in a different direction. And so I think your kind of comment there is, well taken. That ultimately doing these kind of like so yeah, ultimately I think we're looking initially at more additive strategies. Like how can we add function or increase functionality so that, there's competition with these methanogens. I think that's probably the best way to go, right? At least from what I can see right now.
All right, if we don't have anything else, I think we can call it. Yeah. Thank you so much for being here. I can send you a recording if you want.
Sure, that'd be great. Yeah, it was really a pleasure to sit and talk to you guys. So, my email, I think if it's fine to make it available if you guys have any follow-up
Yeah,
questions or if you have a great desire to come and work at UC Berkeley, like we're always looking for graduate students and people who are interested in kind of what we're doing at IGI. So please feel free to follow up.
sweet. All right. Thank you so much, Spencer.
You're very welcome.
Have a
Have a
good
good
night.
one.